So the one that we have discussed is basic, right? The one that we have discussed the last lecture is basic, that you have to understand on how you get the absorbance as well as absorbance and emission in fluorescence technique. Absorbance in UV visible, whereas absorbance and excitation and emission in fluorescent signal. Okay, so these techniques is very largely being used in development of optical biosensor. So when we talk about optical, we are talking about lights, we are talking about uh, color change, we are talking about uh, reflectance, all sorts of optical related signal. Okay, all right. This is basic, all right. The one that we have discussed from the beginning, which is the interaction between analytes and receptors. So receptors, we have discussed receptors, a lot of receptors starting from DNA, antibody, aptamer, okay? So all this can be integrate in the optical technique. So optical is here, okay, as the transducer. So what is the purpose of transducer? Is to translate, yeah, to translate the interaction between analyte, the one that we wish to detect, and the receptors. Receptors can be biological as well as it can be chemical reagents. Yeah, all right. So remember, transducer is to the one translates the interaction of analytes and receptors into a signal. It can be, if it is electrochemical, we observe the current. We have discussed about techniques in electrochemical. So in optical, we can observe color change. We can observe a refractive index. We can observe other characteristics related to lights apart from color change or absorbance or emission. There are a lot of other characteristics related to optical that we can use in development of optical sensor. Okay, all right. So optical means light. So optical transducers are devices that convert any signal, okay, to or from that involve light. So when the analytes interact with the transducer, there is some changes towards the characteristic of that light. Okay, so by observing the changes of characteristic of lights, okay, it might be, as I mentioned just now, it might be color, it might be wavelength, it might be refractive index. So that changes can give us an information on the analyte that we are measuring. Okay, so this is the basic of optical transducers. Okay, sorry, it's a bit blur. Okay, so again, these are among we have discussed last week. Right, let's check. Uh, Shalini cannot hear me. What about the others?
We
Can sorry?
we can hear you, Doctor.
Yeah, okay. All right, so these are the types of uh, machines. Yeah, transducers is optical, but these are the types of machines related to optical technique. So last 2 weeks we have discussed about UV visible spectrophotometer as well as fluorescence. Yeah, but there are others like SPR, okay, and then optical fiber. So there are others as well. And as I mentioned, for optical sensors, okay, they use signal like refractive index, absorbance, fluorescence. There is also called Raman scattering. So these are characteristic of lights that we can play around to get the information of for the analytes based on the uh, interaction of these analytes and the receptors. Yeah. So these are the things, but I'm not going to discuss detail on all the different techniques of optics because not all is in my expertise. All right. Okay. The first that we have talked about so many times is on color change, and it is based on naked eye. Naked eye means that you can observe the changes just by looking at the color change uh, when you test for example, your blood or your saliva. Okay, so this is well-known test strips, rapid test kit, so you can observe um, the color change, whether you have two lines, control line and test line, or you might have only one line, which is the control line. So these are example of optical based sensors, and the color change is due to gold nanoparticle. Yeah, the color change is due to gold nanoparticles. So the color that you observe is actually gold nanoparticles. All right. There's a lot of paper-based uh, naked eye color change that you can observe. Apart from gold nanoparticle, there's also enzyme. Okay, sorry again about the it's a bit blur, but you don't have to worry about this. So these are color change based on reaction of enzyme. Yeah, this is we have I think glucose oxidase GOX here, and then when it react with glucose, okay, there is a chemical reaction between okay, once the glucose being oxidized by glucose oxidase. This is an enzyme specifically oxidize glucose, and from that reaction, gluconic acid is produced plus peroxide H2O2. So if we add TMB, I don't remember the full name of TMB, it's a chemical reagent. And if there is peroxide in the system here, uh, there is, and also another enzyme which is called peroxidase, hydrogen peroxidase. This will uh, produce colored form of TMB. Okay, so there is a change of color from original TMB to another form of TMB. It's not clear to me, but this is actually the principle of the glucose meter. If any of you has used glucose meter, or you know someone else is using, that you can buy from the pharmacy. So this is basically the principle of how it works. Okay, on the strips that we put our finger finger prick drop a blood. Okay, here. Okay, so the glucose in the blood will react with the enzyme, and there is a series of another reaction. There are two enzyme involved here, and the intensity of the color produced is going to tell you about how much glucose is in the sample. So you can see actually it's a simple strip, but it gives you information. So if you are familiar with the glucose tester, you have to drop your blood here, and there is a capillary effect that move your bloods towards the sensing area. Okay, this is another example of color-based, yeah, color-based reaction. So ELISA, the full name is immunoassay. Okay, there is a full, it's an immunoassay and it is based on antibody. So we have discussed a lot about antibody structure and how it has the specificity towards the analyte. Okay, so we can design. Okay, we can design the reaction to have colored base. So in this case, we can design it to have like blue color is positive and pink color means it's negative. So we can do that. We can do the chemistry to have a different color for positive reading and negative reading. So these are all based on naked eye. And there is a material called quantum dot. Yeah. So just now, if you are familiar with rapid test kit, most of the rapid test kit, they are using gold nanoparticle, okay, to produce the color. This is another material called quantum dot that can also produce colors, but it requires uh, fluorescence resources. You cannot just look at it uh, in in normal daylight or normal lights, but it has to be under fluorescence lamp. So if you look at it under normal lights, you cannot see the colors. Like here, we have different sets of colors uh, due to different size of the nano quantum dots, yeah, nanoparticles of quantum dots. So if it is under fluorescence light, you can see the difference. So this is another aspect of optical beam sensor where we can use uh, multiple or different kind of nanomaterial to help us producing colors. So just now we have uh, gold nanoparticles. That is quite common. And we also have quantum dots. So this is one example of nanomaterial that can produce colors, but under fluorescent condition, fluorescent lamp. So if we are used to rapid test kit with gold nanoparticles, the quantum dot as well can be used to develop rapid test kit. So you can see here, this is the rapid test kit being developed based on quantum dot. So in order to see the lines, it has to be under UV lamp, under not fluorescent, under UV lamp, so that the fluorescence of the quantum quantum dots can be observed. Okay. Um, the good thing about quantum dots is it is more sensitive compared to the gold nanoparticles, but the difficulty is you have to use UV lamp to look at it, to look at the lines. It cannot be observed by just naked eye. Okay, so it's more sensitive because if you have experience with rapid test kit, sometimes it is not that sensitive. So by changing the material from gold nanoparticle to quantum dots, we can get a better rapid test kit. Okay, so this is a good thing about the nanomaterial. We can do some advancements so we can improve the sensitivity of the rapid test kit. Hmm. So those are the one that with naked eye means that you don't have to use machine. The one with quantum dots, you have to use at least UV lamp. Okay. Next one is portable device. You start the optical sensors using machines, using UV visible spectrophotometer, using fluorescence spectrophotometer. Okay, So of course, in order to have a portable optical sensor, we need to develop also portable UV visible device, portable fluorescence device. So I'm not here to talk about the engineering part of the sensor, but it's for you to understand the principle, how it works, just how the UVs or the fluorescent spectrophotometer being used in development of optical sensor, sorry. Right, so this is an example. If we have portable uh, UV visible spectrophotometer, it's a small device. And instead of a big device, we can use optical fiber. These are all optical fiber that can bombard lights. Okay, okay. Remember about the principle. Okay, light is bombarded. Okay, and then of course, once light is bombarded, uh, the compound, okay, the chemical compound that receive the photon or the lights or the energy will have electron excited, okay, at certain wavelength, right? Okay, all right. And then what happened here is, okay, the compounds which is excited with certain wavelength, okay, will absorb. Once it is absorbed and excited, of course, you can observe the absorbance at certain wavelength, okay, right? And then let me finish this. So what happened here, if we have a very specific transducer, okay, I put here reagent because this is what I did before. I use chemical reagent as the transducer. Okay, so let's say we have different kind of transducer. We have DNA, we have antibody, we have aptamer or any other transducer here. And then we react with the analytes. Okay, we react with the analyte, and then there is a uh, light, okay, that bombarded here, and then there is excitation of electrons, and you will observe a change in terms of, okay, this is, and then you will observe a change of absorbance and emissions in certain wavelengths. So I don't have the examples of spectrum here, okay, but I believe the last lecture that we discussed, there are a lot of examples of absorbance, emissions, for example, study of sunscreen. Okay, so you can check back the last lecture notes. Okay, examples of absorbance and emission spectrum changes once the lights is gone through here, and then in the chemical reagent, usually there are chemical compounds that react with the lights. Okay, so you have the one that we discussed, electron excitation, and then electron dropping down, emit some certain energies, and there are also some fluorescence. So those are the things that happens here. Okay, and it can be quantitatively. Okay, if you look at back, okay, the process happens here, and this is the light source that is supplied to the chemical reagents or biochemical reagents, and there is interaction here, and whatever happened to the optical characteristic just now, whether in the form of lambda, whether in the form of refractive index, whether in the form of frequency, is translated back into what we call as a detector. So this detector will sort of give you, it can give you directly a reading of absorbance, reflectance. Is up to the machines that you buy. Okay, So this is basically a very basic, very basic of optical sensors based on UV visible spectrophotometer, a portable UV spectrophotometer. Okay. Yeah. So remember the reagent here is an example. It can be DNA, it can be antibody, because these chemical reagents serve as serve as receptors. So right. Okay. So that is a UV visible spectrophotometer, but the one which portable one. But there's another one called surface plasma resonance. In short, it is called SPR. So this is also uh, a machine, so device, instrument that can be used in development of optical sensors. So this is another what we call as a characteristic of light. Okay, so if you see here, this is in SPR. This is very um, if we buy, it's a very uh, ex- what expensive machine, yeah? But it's quite accurate. So the sensor surface usually is gold, okay, is gold. So of course there's a bit of chemistry that you have to do. We have learned about the chemistry on how to modify gold surface. So now on the gold surface or the sensor surface, usually SPR they use antibody, okay, specific antibody. So it's up to you. You can also use DNA, any other types of receptors. And once the analyte move here, is the receptors. In this case, antibody can capture the analytes. Okay, and this interaction of analyte and antibody triggers changes of intensity, uh, what you call angle. Okay, I'm not really in this machines. So there is a change of angle. You can see if there is no analytes, the white one, there is different angle. And if there is analyte of interest and being captured by the antibody, there is a shift. Okay, so uh, uh, this will be accepted as a signal change. Yeah. So this is the one that being measured is the angle of shift before and after the reaction. Okay, before and after reaction there is a shift. So this is actually it's a big machine, plasma resonance, but there are some who has tried to miniaturize this one, yeah, miniaturize to make the machines into smaller version. But this is one of the optical based sensor that we can use to develop the optical based sensor. Um, so this is the explanation. Energy carried by photon can be okay, all right. This coupling settled. All right. So what is being measured under SPR is refractive index. Okay, changes in chemical properties within the range of plasma field. For example, just now is interaction between antibody and antigen can cause changes in plasma resonance. So these changes can be measured as a change in the angle of incidence or shift in the wavelength of light absorbed and can be measured as change in the SPR signal. So this is the one that being detected. There is a change in the SPR signal. Okay, so we don't have this machine in our university, so it's difficult for me to show you. Okay, so this is an example. another example of how the refractive index. Yeah, the lights is shown here, so there is a change of angle of reflections after the interaction between the uh, receptors and the light. So that is the principle of SPR. So we started from um, rapid test kit, which is a naked eye. We moved to optical, the first optical, which is UV visible spectrophotometer, as well as fluorescence, and the other one is SPR. So this is another optical sensors. It is based on photonic, so I don't know. The it is called photonic. Okay, photonic still utilize lights. Okay, so you can see that it's wearable, means that you can wear it. Uh, you can wear it. In this case, it is worn on hand. So the lights, okay, yeah, is but there is an implant. Okay, there is an implant inside the body, inside hand. Okay, so there is interaction between the lights, okay, bombarded from the system. Okay, there is uh, I'm not sure about the system, but lights is produced and it interact with the implants in the body, and when it goes back, you can see that there is an arrow going back. So there is information, new information, giving to the smart band. Just now the whole thing is called smart band. So the information can be processed and giving us the the new information. For example, I'm not sure about implant here. Yeah, I think uh, it is up to what we want to measure. If the implant is measuring glucose, then it should be giving information on glucose. If it is measuring something else, then it must be giving um, information on something else. But smartwatch, we do not have implants. So example of oxygen level that we discussed from the beginning, I think it's just an interaction between the probably the photon with the oxygen in the blood. So I don't think that, but here is quite specific because uh, there is an implant already in the body, and the photon from the smart band is interacting the implant to give the information that the machines want. Okay, all right. So next one is the material. Okay, there are some material, specific material that is being used in optical based sensors. Just now we talked about gold nanoparticles. Yes, there is one. And we can also use silver nanoparticles. Yeah, silver nanoparticles also can give us color change instead of uh, apart from gold nanoparticle, there is silver nanoparticles. And just now I mentioned quantum dots, and there is also called carbon dots. Okay, so what's the difference between quantum dots and carbon dots is the starting material. So carbon dots, the starting material is carbon, whereas quantum dots, there are a mix of oxides. Yeah, I'm not going to talk about quantum dots here, but it is not based on carbon. Okay. And there is another types of quantum dots called graphene quantum dots. So the quantum dots is based on graphene, right? And I skip the molecular beacons. Okay. This is still um, being used. Yeah. So what's the difference? What is actually molecular beacon? So you can see that you have the ATCG, right? So it's a series of nucleic acid. It's a series of nucleic acid, and it is based on interaction of ATCG. So we have learned about the reaction between A, T, C, and G. Okay, so molecular beacon, the one that they modify is what we call as colored dye. Yeah, you have CY5 and CY3 as fluorescent dyes. So they are actually colored in fluorescence, yeah, in fluorescent environment. You can see the color change. Yeah, these molecular beacons is being used a lot in PCR, yeah? Expensive PCR. Um, so how does it work? Okay, if there is no matching, you can see if there is no matching DNA, if there is no matching DNA, the shape of molecular beacon will stay as as shown here. That's why it is called beacons. Okay, and the fluorescent dye here, okay, is in very close proximity between each other, so CY5 and CY3. So when they are very close together, there is what we call as fluorescent resonance energy transfer. In short, it's called FRET. Okay, so FRET can be measured in a form of fluorescent signal. So you can measure the FRET. So let's say if we exposed an extracted DNA from the sample, and the sample, the extracted DNA is a match. Okay, it's a match with the molecular beacon, and you can see that the shape of this molecular beacon spring, you know, open up. I was saying a spring up. Okay, open up because this there is a match between ATCG of the molecular beacon and the extracted DNA. When they open up, you can see that the position of CY3 and CY5 now is far away between each other. So due to this, uh, the the FRET signal is no longer observed after the interaction. So what is molecular beacons? It's actually a DNA probe. Okay, if you remember DNA probe is the one that specifically designed specifically designed. Uh, you remember DNA probe specifically designed to that particular species. For example, if you want to detect E. coli, then we have DNA probe for E. coli. So same goes to molecular beacon. It's actually a DNA probe specifically designed for that particular target. Okay, but it is in the form of beacon, specific form in this structure, and tagged. They are tagged with fluorescent dyes. Okay, so once they find the target, okay, the right target, for example, then the structure of the beacon open up, and due to the separation of the fluorescent dyes, then the FRET signal is no longer observed. So what we can conclude from this, if there is energy transfer, FRET means that uh, there is no target DNA, or there is no species of interest in that sample. So if there is FRET, if there is no FRET, means that this is happening. So there is target of interest in the sample. So this is what molecular beacons. Okay, so this is one of the example of optical based material that we can use in development of optical sensors. Yeah. Uh, so advantages of optical sensors is actually relative. Sometimes it's difficult because it is colored based. So if your sample is colored based, then it's going to have an interference. You have to do cleaning up, separation first before doing the analysis by using the optical sensors. Of course, this is all something relative, whether you uh, if you use electrochemical, there is some disadvantages. If you use optical sensors, there is disadvantages as well. So this is relative, right? Okay, so this is all about optical sensors. If you have any question before we go a bit on the ENOS. Or you want to discuss ENOS after the 8 to 9? Maybe you want to take a rest first?
It's okay as you want. Yeah.
Sorry?
So
Yeah?
both is
Both okay. All right, okay. Just a moment. I'm going to change to the ENOS. Okay, if we finish early, means that you don't have to do 8 to 9 because today is only these two slides and we can finish. I think we can have extra like 7:15 so we can finish all the ENOS together. So we don't have to do the 8 to 9. Okay, for all of you?
sounds good, Prof.
Yeah, all right. Because this is actually supposed to be delivered by Dr. Amar, but now, yeah, he's a bit busy. But if you want to have the detail, you can watch his recorded video. There are three parts. There are three parts of his recorded videos. So what is actually ENOS? Very interesting. Okay. Oh, very interesting technology. Okay. So we have learned about electrochemical, optical. So this is actually, uh, it can be for ENOS, it can be optical as well. It can be electrochemical as well. We'll see. Okay, so this is one of the that we have developed together with Otama. Still is very big. It should be smaller. So what is ENOS? Well, it is something that you can imagine that you human nose. We sniff. Okay, we can sniff things, and then we can identify like this is the smell of coffee. This is the smell of chocolate. So it works like that. But instead of um, it's a artificial nose. So how does artificial nose being developed? Okay, this is something that quite trendy because due to COVID-19, there is a need, yeah, to to mod to actually monitor and to check, especially at airports. They need to quickly check whether there is cases of COVID or any other respiratory diseases. Okay, so there is during the COVID-19, I think there is one airport that is using dog, yeah, to sniff the people arriving, okay, who actually might have COVID-19 and whatnot. So this is very interesting. And a lot of this enos is used in gas sensor technology. Okay, for example, there is leaking of ammonia. Okay, we contaminants, and if you have air purifier at your home, yeah, air purifier, for example, from Dyson, from Cowie. So they have this kind of technology in the system. Okay, probably not too specific. For example, it is not really specific to formaldehyde. It is not specifically to ammonia, but they claim it is quite specific. Probably it is not, but it's a good technology because if it is, for example, if it is ammonia detected in high level of close confined spaces, then it is very dangerous. No? So if the reading of ammonia overshoot, then we can have an alert system asking the people to evacuate. So this is what uh, ENOS is very interesting. Okay. All right. So what we will cover, just some simple, because this is not my expertise, but I work together with Dr. Amal and his team. So I do understand a bit on how the ENOS work. So so this is a picture of example of ENOS. So ENOS consists of an array of gas sensors with different selectivity. Uh, this one, okay, you can see that small, this different array of gas sensors. And it is by design. You can put as many as you want, as long as you have a good system to discriminate between the signal. Um, so one thing about ENOS, it has to be with pattern recognition. Okay, so I will discuss why. Uh, right, let me skip that one. Okay, this is an example of ENOS. This is application of ENOS. You can use ENOS, of course, to diagnose diseases, especially respiratory based. Yeah? So when you breathe in, breathe out, certain chemicals being expelled out, and then it can be exposed to the sensors, and the sensors can do some analysis, and the reading can be translated into whether there is a possibility of you have certain disease. There is a company in UK who has developed, I think, a quite established ENO system for lung cancer. Okay, and the story is very sad because the one that developed this ENOS company, he has a wife who died due to lung cancer. So I think he has this motivation to build an ENOS company for lung cancer detection. I think is actually progressing well, the ENOS. Yeah? And the ENOS also can be used to check the quality of food. Yeah, because as we know, when certain food goes bad, not if it is spoiled, if the food is spoiled, of course there is some uh, bad smell. Okay, but we don't want uh, to know to actually up to that stage until it is become smelly or whatsoever. We need to detect uh, before it is smelly, before the food is smelly. Because for human, we can only smell, okay, when the food is smelly, then we know it is spoiled already. But for machines, they can detect early. So we can train the enos to detect, because we can train, or we can develop the enos to be sensitive towards certain chemicals that being produced by the spoiled food. Okay, so at least we can have um, that kind of sense. So, for example, uh, in packaging, so you can at least cannot don't have, what do you call, sometimes you buy something but it's actually already spoiled and you lose your money. So we don't want that to happen. So probably certain companies, food companies, should employ this. Yeah? And not just for spoilage, okay? Enos as well can be used to for what we call quality control. Yeah, because if you right, if you know that, for example, certain companies like wine companies, I think coffee companies as well, they have to train someone to smell the wine, to smell the coffee, to know which one is of good quality, which one is not. Okay, there is so this is crucial, but it is still based on human, and it's not. We don't have that capacity to have that many experts in that in that kind of area. So if we can have a machines that can be trained to uh, take care of the quality, to be trained to learn on the quality control of the food product, then at least we can have more rather than 1 or 2 person who have the expertise. Okay, so this is something can be explored. All right. Same goes to fragrance, perfumes, flavor. And I work in agriculture. What I have developed with Dotama is enos to detect a fungal called uh, fungal infection in palm tree, oil palm tree. So it is a big issue. Ganoderma boninens is a big issue. If it is infecting, if it is uh, infecting the tree, then the production of the palm tree, the fruits and everything will be reduced greatly. So the important thing is to have an early detection when the fungus is actually inside the tree. So the enos should be able to sniff the infection by exposing it to the leaf of the palm tree. Because if the fungus is already out there at the trunk, then it is too late. Okay. Right, so I'm not going to discuss about the money. There's a lot of money. So this is a comparison between enos and human nose. Okay, so for human, we have receptors as well in our nose, and the brain will analyze whatever information the receptors giving to the brain, and you have a conclusion. Okay, this is coffee, this is serai, this is daun kesum. All right. And same goes to gas sensor or enos, okay, in short, because we are dealing with volatile chemicals. If you have a lot of sensors in which is mimic the receptors in the nose, and there is processing. Part of it is processing, for example, pattern recognition. There's a lot of processing software that needs to be done when we work with enos, and we can finally conclude same way as our brain concludes the final one. Okay, this is what this is the smell of. For in this case, we have mold, we have bacteria, because most of it they give out certain volatile chemicals. All right, so let me uh, okay explain to you the different types of gas sensor technology. As I mentioned, it also can be electrochemical, it can be optical, because based on the principle being used, or we can also have field effect piezoelectric. This is something that I haven't discussed with you, but at least you get to know this in enos. Yeah. So this one I skip because I really don't have the knowledge on this. This is what we call FET, field effect transistor. Okay, field effect transistor. So this is more towards engineering, towards physics. So there is a change of resistance. Yeah, here there is a change of resistance if the material here being exposed to certain volatile compound. So you can see here voltage, ampere. So there is a change of resistance, voltage as well as uh, ampere, or in this case current. Okay, so the basic is simple. If it is exposed to this field, in this case, the example is ammonia, we can observe some changes in potential, voltage or potential, and current, as well as resistance. So that is the basic. And this is semiconductor. Okay, so semiconductor, it is a lot relies on free electrons. So we have the semiconductor material here. Also, this is not my field. When you have a volatile, in this case gases, it depends whether you have reducing gas. Most of the time it is reducing gas, and it will trigger movement of electrons. Okay, so the movement of electrons will be detected by uh, uh, this these machines. Okay, in this case, for example, this is example of the sensing material, where we put semiconductor here. This is a semiconductor, and there is free electrons here. Okay, and then when you have reducing gas, it will interact with the oxygen, and there is a movement of electron. It might give away, or it might, okay, in this case, for example, if you have clean air, then you have different electrons. Yeah? So if you need to know more, please do listen to the recorded lecture by Dr. Amar. Okay. All right. So this is piezoelectric. So piezoelectric, it is based on certain material called QCM, quartz crystal microbalance. So if we use the quartz crystal, it's a crystal. It is based on mass. So the mass of the gas interacting with the crystal. So if there is changes of the gas mass interacting with the crystal, it can be detected by this machine. So it is detected based on frequency. So again, I do have this machine, but never use it. So such a waste. I don't know. All right. So this is how it looks like. The quartz crystal electrodes, where actually we can have gold, and the gas is interacting the gold surface. You can add some material uh, if it is sensitive towards, in this case, nitrogen dioxide. And there is a change of mass. You can see delta M, and the change of mass is translated into the change of frequency. So what is recorded in here, the final one is measured is the change of frequency. So this is what the reading looks like. Okay, if there is interaction, you might have a change in the frequency, a drop in the frequency. Okay, so this is basically what QCM, post crystal microbalance, looks like. Okay, so I'm not going to go more, but this is electrochemical. And this is optical base. It is not colored, but they are using lights, photon. Yeah, photon. You can see this is again the sensors, and then when the light is bombarded on the surface of the sensors, it reflected back, and reflected back, and then there is a change of information. So that will be the base of information on gas sensors. So this is an example of the parameters being measured in eNOS or gas sensors. You can see conductivity, fluorescence, piezo just now, piezoelectricity, frequency. Okay, so this is basically if you compare gas chromatography, for example, because gas chromatography is usually the one that we use to analyze volatile compound. Okay, so if we expose volatile compound into gas chromatography, usually if we do not know, we don't have the standard of that sample or the standard of the analyte, we need to run mass spec as well. Okay, but if we have the standard of the analyte, we can just use GC. But for eNOS, so same principle. If we exposed the volatile compound, okay, and then there's a lot of sensors. We need a lot of sensors. And then all these volatile compound interacted with the sensors, whether it's semiconductor, whether it is FET. Okay, and then those signal we have to be analyzed by using pattern recognition, all those kind of PCA. There's a lot of software that you can use to do data analysis before the conclusion can be drawn to check whether you have what kind of volatile compound. Okay, let me give you example. Okay, this is an example. Okay, this is an example, for example, from cultured cell lines. Yeah? So these are all chemicals from cultured cell lines, which is related to cancer. So, so instead of using standard techniques to check whether there is some which chemicals actually, which volatile chemicals actually giving an indicator that you have, for example, lung cancers. Yeah? So we have to study which chemicals. So from there, we can develop an eNOS. Yeah, an eNOS. In this case, I don't know whether it's correct. Okay, the canal, yeah, is observed to have this. I think in A549. This is one of the cultured cell lines, and I do not know the detail. So they found out the canal is it seems to be the indicator. Yeah. And from there we can develop a series of sensors that responds to a certain aldehyde. Okay. And then probably some alcohol. So we can design the sensors to have this responsive to some alcohol and aldehydes. And from there, we can try to do some pattern recognition and so on. This is not easy, as you see. It's difficult, okay? From this, we can try start to develop an e-nose, for example, to check on the cancer, probably the lung cancer, probably, okay? So that is the basic of how e-nose, yeah? So you can go read all this. And please do listen to Dr. Amar's talk. There are 3 parts which I cannot cover, but this is the basic. This is a very basic that I can relate to you. And if you're interested, we are still doing some e-nose sensing in our lab. So if any one of you are interested, can come and check how we do it and how the e-nose look like. Okay, all right. So this is some of the results, and this is some pattern recognition we do to identify the chemical compound and the concentration. All right. So Dr. Amar's company right now is doing something on air quality for hospitals. We work as his partner, doing all the wet chemistry lab. Okay, probably one day you'll find our air purifier, an air purifier attached with our system, probably one day. All right, thank you so much. I think we do not have to continue on 8 to 9. So next week, I have invited Dr. Helmi to talk about enzyme because enzyme is not my expertise. So he'll be talking about enzyme, one of the receptors. Enzyme is a very good receptor, and he'll be talking about examples of enzyme-based sensors. His PhD is in enzyme-based sensors, and he's from Faculty of Biotechnology. Okay, so any questions? Everything is good with the assignment? Any question on assignment?
I think no, Doctor. Thank you.
All right, thank you.
Thank you
Okay,
so
so
much.
all right, it's towards the end. There is another 3 or 4 weeks of your lecture. Okay, so next week will be Dr. Helmi. I will start the class and Dr. Helmi will come in. All right. If there is no questions, then thank you so much. Okay. Have a nice break. Bye-bye, everybody.
Thank you, Doctor. Goodbye.
Good, Doctor.